Did you all get the message? Are we live already?
Yeah, it says meeting is now streamed live on YouTube. Yeah.
Okay.
Is it live?
Yes, it's live. We're good.
Yeah. Okay. So, welcome everyone to this seminar today. My name is Adriana Maldonado Chaparro. I'm a professor at Universidad de Rosario in Colombia, and I'm your host for today's seminar. And I'm really happy to have you all here. So before we start with today's seminar, I want to thank our previous speaker, Peter Boston, for his awesome talk on social evolution in anemone fishes. And I want to invite you all for our next seminar. David Queller will be talking about cheating in a social microbiome. So it's going to be a little bit of a shift in our study system. For today's seminar, we have Josh Firth. Josh, he got his PhD at University of Oxford in 2016, and since then he's been a fellow researcher at different universities, Oxford and Reckitt. Currently, he holds an independent fellowship at University of Oxford as well, and his research focuses mainly on understanding how ecology and individual behavior interact to shape both social structure and the consequences of social processes, specifically of contagions, both in human and animal societies. Josh has an impressive record securing funding for his research, and I'm just going to point out two of the most recent ones, an NERC, Independent Research Fellowship, and a Wild AI, large research grant that I'm assuming it's using for funding his current research as a PI. His publication record is very diverse and reflects the great contribution that he does to our area. He publishes in a wide range of journals such as Nature, Ecology and Evolution, Behavioural Ecology, Journal of Animal Ecology, plus Computational Biology and Methods, and many others of these. But he doesn't just stay within the scientific realm. He's also very active communicating science through podcasts and interviews. So you can check out his website to all that he does out of science. So I don't want to keep you more. So with no further ado, today's seminar title is Behavioral Ecology of Contagions, Pandemics, Passerines, and Permuted Populations. So hopefully we'll hear about our great teats. And Josh, thank you for accepting our invitation. The mic is all yours.
Thanks a lot. That's great. Right, I'll just go straight into it and start sharing my screen. 1 second. Thanks everybody for coming along as well. I really appreciate it. Yeah, we'll definitely be hearing a lot about great tits. Don't worry about that. Okay, so is that on the full size sort of view now? Brilliant. Great. So yeah, thanks a lot for that generous welcome, Adriana, and thanks for inviting me to come and talk today. I've watched quite a few of the fine seminars myself now, so I'm a fan, and it's great to see all the different diversity of topics that I've covered. And today I'll be talking about what I've titled the behavioral ecology of contagions, pandemics, passerines, and permuted populations. I'll be touching on each of those three things. Chosen name mainly for the alliteration factor, but I will be diving into each of those as we go along. So just as an introduction, yeah, thanks a lot for inviting me, but also beware, I've got a broad Yorkshire accent, which when I moved to Oxford 11 years ago now, everyone said you're going to lose your broad Yorkshire accent, but I definitely haven't lost my broad Yorkshire accent. So hopefully I'll still be able to get the main messages across. And yeah, I've tried all different sort of AI translation tools, but they cover every different language in the world so far, but not quite the Yorkshire accent yet. And then in terms of the actual talk, first of all, I'll introduce why I think behavioral ecology might be a useful approach for studying and understanding contagions in general. Then I'll look at a real world case study looking at a disease, a little known disease called COVID-19, and how that might spread through real world social systems. And then we'll dive into some real science with the great tits, of course, looking at how animal populations might be useful model systems for thinking how behaviors in particular might spread through populations. And then I'll highlight some questions at the end, some big questions with no answers to these questions, but just some highlights of some areas which I think might be of particular interest when we're thinking about how behavioural ecology and studying contagions might combine together in future as well. So to begin with, what's the point in even trying to combine behavioural ecology into studying contagions? Why would we even bother? Well, when you first think of a contagion, most people think of this, especially since the COVID-19 pandemic, some sort of virus entering the social network and then spreading through social contacts between individuals. But contagions can actually refer to any process by which entities spread through a social network. And then behavioural ecology, why can that be useful when we think about it as how individuals might respond? What's the evolutionary basis for why individuals might respond to different ecological settings? Well, first of all, behavioural ecology provides a great framework for understanding behavioural variation, particularly in terms of individual level differences in behavioural variation. And when we think about individual level differences in social behaviour, for instance, that's what forms the social network that a contagion might spread on. So here, for instance, just a really simple toy example where you've got two types of individuals: social individuals, the red ones there, and then asocial individuals or less social individuals, which are the blue ones. And you quickly see that the number of ties an individual has, so their own behavioural phenotype, shapes where they might be positioned in that network and how many connections they've got. But behavioural ecology is also good for thinking about not just individual variation within a particular system, but also how variation between systems, either populations or different species, might shape that population in terms of a population level outcome. And then what I've done here again on the right hand side there is just a really simple toy example where we've got a similar network structure with the same number of individuals and then the same type of individuals, but then change the composition of that. And you quickly see that that variation on that level as well. The number of social individuals compared to less social individuals is going to shape that social network. And of course, that's going to affect contagion propensity. So if you introduce some sort of contagion into that network, we're going to see a massive consequence of both individual level variation, so how likely individuals are to become infected or informed from that contagion, but then also population level variation, so the shape of that group and how it affects how that contagion spreads. So that's behavioural variation, but behavioural ecology also provides a brilliant framework for thinking about trade-offs. And trade-offs are very important when you're thinking about contagions, because even though we're all well suited to thinking about the disadvantages of contagions, especially when we call it contagion and think about a virus, there's also lots of benefits of social contagions and benefits of social contact more generally, such as helpful behaviours or innovations might spread through the network, which need to be traded off against disease. So you can't just be totally asocial because you won't get any of the benefits of social contact or the benefits of social contagions. And that's where behavioural ecology comes in, to think about these individual level trade-offs, how individuals might make decisions about that trade-off, but then also how those decisions might be resolved across different systems under different ecological settings as well. And what that might mean for both individual strategies, what the group ends up doing, and then how the population as a whole looks in terms of the social system. And then finally, behavioural ecology is also sort of a traditional framework for thinking about social decisions specifically and how individuals might make changes based on their social environment. So also for thinking about behavioural transmission, for instance, if we just think of a simple contagion, so I'll be referring to two types of contagion in this talk: simple contagions and complex contagions. If we think about a simple contagion just refers to what we think about as maybe how a virus or a disease had spread, where the more ties you've got to infected individuals, the more likely you are to then become infected yourself. So we can see here in this toy example, this individual here in the middle has got the most ties to infected individuals at this time point, so they're the most likely to become infected. A complex contagion refers to some sort of social rule over and above the contagion as well that might be playing a part, where, for instance, a very simple social rule might be something like conformity, where you might say, oh, it's actually not just the total number of ties to infected individuals, but the proportion of your ties to infected individuals. So you can imagine this taking action in contexts where you might have an individual with 100 ties, say, and 10 of those are to infected individuals. In a simple contagion, they're going to become much more likely to be infected quicker compared to an individual with just 2 ties with 1 connection to an infected individual. Whilst on the other hand, if you think about a complex contagion, that same individual again, individual A with 100 ties and 10 of them to infected individuals, only has 10% of their ties to informed or infected individuals. So they've actually got a lower proportion of ties compared to an individual B with just 2 ties, where 50% of them, 1 of them is to an infected individual. So we see there's a large difference in simple contagion and complex contagions and how that might spread through a network. And just to show that as a visualization here, just taking those simple 2 rules. So initially we've got a simple contagion spreading through that network, where at each time point, the individual who's next infected is the one with the most number of ties to infected individual, compared to if we had a complex contagion with the same start point. So this is the red contagion now. You can see it flows in a completely different direction to the way that the simple contagion flows, despite that they've both got the same start points and flowing on the same network. So we can often see in simple contagions, the most central individual is most likely to become infected, whilst in complex contagions, the most central can sometimes be the least likely to be infected or informed. So taking those 3 things, behavioral variation, trade-offs, and social dynamics decisions, and trying to understand them in the context of behavioral ecology is why I think behavioral ecology might be useful for understanding contagions. So I'm going to keep drawing on each of those 3 topics as I go through this talk, when we start talking about some real data and some real case study. So hopefully that's convinced everybody from what's the point in even trying to combine behavioral ecology and contagions. Everyone's sold now, and from here on, everyone's ready for what comes next. So how we might try and understand COVID-19 in real-world social systems. I just found this on Reddit and it made me laugh, and I was guilty of this as well. So this is just a figure saying what gives people power, money, status, and playing epidemiologist once the COVID pandemic kicked off. And yeah, I got guilty in getting drawn into this. So in 2020, as the COVID-19 pandemic kicked off in the UK in particular, the Royal Society put out a call for rapid assistance in trying to understand how COVID-19 might spread in real-world networks. And whilst in terms of studying human real-world networks have been fairly limited until that point, due to the restrictions on gathering lots of data about lots of different human populations, behavioral ecologists had already been studying animal social networks and real-world social networks for a long time. Even at that point, there was over 800 social networks of animals published already. So me and a collaborator, Lewis Spurgeon, who's also a behavioral ecologist, was drawn into helping out with modelling the COVID-19 spread and trying to think about how that can be used for informing SPI-M in the UK, which was the government's sort of advisory body for these modelling predictions. And what me and Lewis in particular wanted to do is try and bring in these principles of behavioral ecology, trying to understand behavioral variation between individuals, how individuals might be making trade-offs, or what that might mean at the population level, and what sort of social dynamics might be at play when we're thinking about modeling COVID-19 spread in these networks. So the basic principle of this was, first of all, we needed some human tracking data, and we used quite a few different data sets, but this first example I'm going to draw on is actually using a data set gathered from a BBT4 initiative of GPS tracking of over 1,000 individuals in a particular town, Hazelmere in the UK. And then once we had that tracking data, we could build some social networks out of it. So then you can build your real world social network based on the tracking data, which individuals are coming into contact with which other individuals, where you've got your points as individuals and then lines between them showing some sort of social contact between those individuals. And then we can draw on the epidemiologists at that point. So at that point, we said to the real epidemiologists, okay, so what do we know about COVID-19 so far in terms of once you've got a social contact, what's the chance of it spreading COVID-19? Once you get COVID, how long until you become infectious? How long until you're no longer infectious? What's your chance of becoming infectious at all? Taking all those different dynamics about the actual particular disease, and then we can simulate that on top of the network to say, okay, how might this disease, given everything we know about it, will spread on this real world network. And then we can estimate things like spread rates and which individuals might be key in spreading this contagion. And one thing that came out at that point, which were already becoming obvious in the UK, was that uncontrolled contagions of this type would spread rapidly in human social networks. But then what we could also do is we could start simulating controls over the top of it, even before the UK had started putting these measures in, and saying, okay, what about if we overlay something like contact tracing, where we try and say, okay, we've got this network, this real world network, we've got everything we know about COVID at this point. How might something like contact tracing, where we isolate the contacts of those individuals we've known have become infected, help stop this disease from spreading as fast? And we can assess the effectiveness of these sort of approaches. And we can also start thinking about these trade-offs again to say, well, primary contact tracing seems like it works quite well. So locking down individuals' friends when they become infected themselves seems like it would disrupt this particular type of infection. While secondary contact tracing, which is not just locking down their friends, but their friends of friends, so going further out into the network there. Also, it still does work in terms of controlling the disease, but it's overly disruptive in terms of actually cutting out too many social contacts for the increased effectiveness of this approach. And then this is where we could also take another approach, mainly developed in behavioral ecology as well, of permuting populations, and say, how can things like social distancing help, given we've got these network structures and this type of disease? So what we were doing here is we were taking the social network itself and then permuting it in a way to say, okay, let's take some of these social ties that we observe and redirect them towards the household, for instance, rather than being outside of the household. What would happen if people spent less time outside of the household, more time inside of the household? And what would these social networks, first of all, look like? And then what might that do to the actual spread of this particular disease? And what we show is that even a relatively small amount of distancing helps control. So you can have quite a big effect fairly fast with just a small amount of social distancing, even if it's redirected in towards the household. Whilst as you get towards more and more social distancing, you might be going into that trade-off of too much social disruption for not enough effect on the actual contagion itself. And we could also use permutations of populations in a different way. So we could take the real world network structure and then start saying things like, okay, pretend we don't have all the information about all these social ties that are taking place. We've just got the information regarding the number of individuals in that network and the amount of time social each of those individuals are having in that network. And in that way, we can start using generative models to say, right, based on that number of individuals social, or this variation in amount of time social, what might different social networks look like? And the importance of doing this was, for a lot of populations, you might not always have the actual exact contact data, or you might not want to gather that exact data, or you might not want to use it for whatever reason. But what you do have is some very simple metrics. Can we generate models from this of a social network? And then when we do that, which a lot of people were doing at the time, how realistic is it in terms of how this disease might spread on those networks? And really what this approach did in these sorts of permutations was it gave a bit of a word of warning that knowing the real social behavior as well as you can is actually very important, because just simulating social networks, even if you've got quite a lot of information on the number of individuals and the total time spent social, things like that, can really under or overestimate the spread rates, but more importantly, alter which predictions you might think alter which control measures you might predict would be the best at controlling that particular disease. So the message coming out of that is real world social behavior is quite important. And the other cool thing about computational approaches and taking these sort of things in mind is you can combine different controls and permutations to assess all these sort of things simultaneously and come up with the sort of like optimal trade-off. I'll just play this little video that should start playing now. So you can say, if we've got all these different measures, for instance, across the top here, we've got isolation or case isolation or primary tracing, and we've got testing or no testing, distancing or no distancing. We can plug all those different combinations in together and then try and say, okay, what's the actual optimal strategy in terms of how much disruption we want to cause to this society and, or need to cause to this society to limit this disease to this particular extent. And that's the cool part of, we also built a little Shiny app if anyone wants to play around simulating their own outbreaks on the town of Haslemere in the UK. And again, another cool thing about behavioral ecology as well is that the principles developed there can also be applied across contexts. So this is what we've done. So it's not just that database, but we've started using a few other databases as well. For instance, Singapore was very interested in what they called cruises to nowhere, where instead of having people going back and forth to different countries, which is obviously problematic at the time of a pandemic, people just have a holiday on a cruise where they go onto the cruise, sail around, and then come back. But even within that, you might be worried about outbreaks on the actual cruise itself. So we got some really high-res data from there, looking at Bluetooth trackers, where you've got not only individuals' contacts with each other, but also their position on the actual ship itself as well. And then we can look at things like how different types of social relationship might shape the social network as a whole and what that might mean. So we know that straight away when we looked at this data, we saw quite strong social structuring in terms of crew are normally associated with crew and guests are normally associated with guests more. One of the more surprising findings was that you might initially expect the crew members to be fairly well connected due to their jobs and moving around different groups, but in almost all cases, whether you looked at close contacts, casual contacts, or any sort of contacts that you could quantify, it turned out that the passengers were more social than the crew. And then you could also identify what spatial factors might be important to this particular contagion as well. So by taking these real world networks and simulating these outbreaks on top of them, we could say, oh, it's actually the food and beverage context, so the dining settings, where in every sense of the different types of social contacts, these are the hotspots of social contact, where you're also likely to have the hotspots of outbreaks as well. And so, yeah, taking the idea that we can apply these sort of models and findings across different populations and across groups, once you've got that, we've been applying it to how COVID-19 or different diseases might spread in workplace networks and in school settings as well, and trying to think about what, in each case, what's the different sort of optimal strategies for controlling the disease without causing too much disruption to the social structure of that population. So that were a quick whirl through how we use behavioral ecology for thinking about controlling COVID-19. And now I'm going to go on to the meat of the talk, which is looking at thinking about behavioral ecology in detail in its own context of animal social networks, and how this might tell us about how behaviors rather than diseases might spread through these populations. And again, the reason why the animal population is really cool is, even though you can use the principles of behavioral ecology to think about how something like COVID-19 might spread on human social networks, you can never really sort of back calculate into, oh, well, what does this mean for advancing these topics in behavioral ecology? Can we learn a bit more about behavioral variation or the consequences of behavioral variation per se, or how individuals actually make trade-offs? So by using animal populations, we can actually test these processes, which I'm going to go into a bit more detail soon, and actually learn about these concepts rather than just applying them. So if you want to build this sort of deep understanding of the behavioral ecology of contagions, rather than just using a few human networks, ideally what you need is two things. So first of all, you need some really large scale data where you've got lots of individuals, hopefully the entire population, and you've got these fine scale details of social interactions between those individuals. And then also importantly, if you're interested in understanding how things like ecology, for instance, or evolution might affect these processes, then you need these things across settings, so different settings. So you've got understanding in different ecologies, and also across generations. So hopefully you can try and predict what might be the fitness consequences of these social decisions, and how does that transpire over different generations. And also, again, another cool thing about animal populations is that you can actually use experiments, which you can't really do in humans, even though I'm sure places like Facebook will try. There's restrictions on changing individual social behavior, but in animal populations, we've got this opportunity to try and manipulate individuals' social phenotypes, to try and manipulate the social structure as a whole, and then actually directly test the consequences for social contagions within these social networks. And where can we find this? As I said, wild animal social systems seem to be the best place to do this. On one side, we've got the lots of different groups around the world collecting social network data on lots of different species of animals. And then on the other side, we've got some intensely studied systems where people are either doing really intense recording on those particular ones across generations, or looking at using experiments to actually test these social processes in real world populations as well. So what I'm going to do now is I'm going to use our great system at White and Woods to first of all talk you through one example of a model system, which is our wild birds in White and Woods. And then secondly, how we use intensive recording within this population to try and untangle some of these processes to do with social contagion a little bit more. And then third, looking at how we also use experiments on this wild bird population to actually test some of these predictions. So first of all, the model system at White and Woods, which was, so it's David Lack who started studying this system right back in 1947, where he was studying the breeding birds. So every spring going around monitoring the nest boxes in White and Woods. And now this breeding data set is massive. So over 20,000 breeding attempts by these birds studied. We've got 1,000 nest boxes through Whiteham. And just to give a bit like of a background of the sort of data we get from that breeding data set, you can look at the number of birds we've rung, which is over 100,000 birds now ringed since 1947. Nearly 150,000 eggs laid in the past 75 years that have all been monitored, of course, then going forward. And then you can pull out some cool facts as well, like the most eggs by a single female. One female laid 74 eggs over the course of her life. One bird had 67 offspring, so that's some serious lifetime reproductive success there. Some birds live as old as 9, and you can also trace individuals back 35 generations, which is obviously like thousands of years for. If you wanted to do that with a human, it's equivalent to 1,000 years, but you can go back great granddad of a lot of these birds, sort of thing. So some really cool sort of breeding data. That's where you get your cross-generational power. But then since 2007, not just studying the breeding attempts, but also studying these birds' social networks as they break down from, so in the spring, they've got these tight sort of territorial pair bonds, social structure, but in the winter, they form these foraging flocks and move around the woodland together looking for food. So since 2007, we've been working on studying these birds' social behavior during the winter as well. And the way that we do this is we've got 60, so the baseline data comes from 65 feeding stations throughout the woodland, which were turned on every weekend. So you get a snapshot of social structure. So you know where individuals are because we've put a tag on each of the birds' legs. So these have got a pit tag that's detected on these RFID antennas. So we know which birds are going to which feeding stations, what time they're there, and then also importantly, which other birds they're there with. So who they're there with in the flock. So to show you what this looks like, there might be a little bit of background noise. It's just going on at the same time with the feeder. The birds come in, it reads their pit tag because they form in sort of flocks. They'll read their pit tag, and then we know which birds are foraging in flocks together. And we get their record like that. The cages around are just to protect from squirrels and things like that. And then this is where you get into some serious intensive recording, because if you look since 2008, say, to 2017, more than 35 million of those detections you were just seeing up on the screen just then of more than 10,000 individual birds, and then these massive networks that you make from this of nearly 1 million flocking events and over 5 million flock memberships where we can assign individuals to particular flocks, and then lots of social network links. And then in terms of just associations of individuals being linked at some point to another individual, up to 27 million associations between individuals that we can start to then use to build a social network to say which individuals are connected to who and when. And that's where we get our intensive recording. So I'm just going to give a couple of examples of how we can use this sort of intensively studied population for trying to understand behavioural contagions more generally. So one question is, how might we understand the diffusion of a new solution in terms of where it starts in that network, in terms of who might be the inventor? So if you're getting a new innovation, who's the inventor, and what does that mean for how that innovation spreads through a social system? So for instance, if a highly social individual comes up with a new idea, they're likely to have lots of social ties, and that if they're the problem solver, then that problem solution, the solution to that problem is going to spread through the network fairly fast. Whilst if someone more peripheral, with less social ties, less friends, if I come up with a new idea, it's not going to spread very fast because I've got so few social ties anyway, no one's going to listen, and it's not going to get out into the network. So then one of the questions you might want to ask is, how might an individual's social position within the network in terms of where the solutions start, how might that affect the spread of that solution? So to do this, we combined it with data we've got on these particular birds about their problem-solving abilities. So first of all, we want a simple problem-solving assay, which is what Ecology did back in 2012, where these birds were taken into captivity just for a short period of time and tested, are they a problem solver or not, using this simple assay where they had to pull a lever out and the waxworms would drop into the bottom of the tray and they get the reward. So whether or not they could solve this simple problem seemed to be unrelated to hunger or neophobia or exploratory behavior. So it seemed like a good sort of independent problem solving tasks. And it was related to other problem solving tasks on smaller subsamples, both in captivity and in the wild as well. So it seemed like a fairly simple assay, even though it probably doesn't capture all aspects of problem solving, a simple assay to carry out on these birds to see whether they're problem solvers or not. And then we wanted to relate their problem solving ability for this brief time in captivity to what they were doing in the wild in terms of where they were positioned in these social networks. And we had a lot of birds done. So there were more than 800 birds assayed over these 3 years, and nearly 500 of the birds turned up in the social network as well. So we know whether or not they're a problem solver and also their specific position within these social networks. So then the question is, how does an individual's problem solving ability relate to where they're positioned in the social network? And then the follow-up question to that would be, and how does that relate to how a new solution, if they do come up with a new solution to a problem, might spread through the social network? So to answer the first part of that question, we found that for the juvenile birds in particular, the problem solvers, so those that could solve that problem in captivity, had a higher degree. So they had more links within the social network compared to the non-solvers. So if you look at the degree of birds here, those problem solvers have a higher degree in general than the non-solvers. However, if you forget about just the raw number of connections and think more about this, the strength of their bonds to the individuals that they are connected to, which we might call average strength. So even though the solvers have more connections, they've actually got weaker connections to those individuals that they are connected to compared to the non-solvers. So the non-solvers have got fewer connections, but quite strong connections. So the friends that they've got are quite good friends. They're regularly hanging around with these, while the solvers seem to be on the opposite side of that, got lots of friends, but not spending as much time with each of those friends. I won't go into this in too much detail, but there's a lot of spatial structuring in our population in terms of where solvers hang out and where social birds hang out. But even after we control for those spatial factors and other things like individual sex and personality, we still see this pattern of the solvers being the ones with lots of connections, but weak connections, while the non-solvers being the ones with fewer connections, but quite strong social connections within that. So problem solvers have got a higher degree, so more links, while those non-solvers have got higher average strength, so they've got higher bond strength. So now we've got those results. What does that mean for how how new solutions might theoretically spread through this population, given that we've got the solvers in the more central positions? And to do this, we take a similar approach to what I presented about with COVID-19. So we choose a start individual. So we know the empirical networks, and then we choose a start individual. So we either choose a solver, a non-solver, or just a random individual within that network. And then we seed them with a simulated piece of information, a solution or an innovation. And then we monitor how that might spread through the empirical networks. So the number of individuals informed at each point in time. So this is what I'm explaining here. So we've got time there on the x-axis, and then the spread rate on the y-axis, where I've plotted it as spread rate compared to random, because obviously a simulated contagion always spreads. So we want to know what's the random rate of spread of that particular contagion, and then how this might differ to that. So we've got time on the x-axis and spread rate compared to random on the y-axis there. So random is obviously just 0. That's what we expect, the spread rate at random. And then we see if we seed a solver in this population, the solution spreads faster than what we'd expect. And this is true for all 3 years. So if we go around seeding solvers, on average, the solutions are going to spread faster than if we seed non-solvers or random individuals. And the opposite of then, of course, true if for whatever reason, one of the individuals we know in real life are non-solvers did come up with a solution of this sort, it would actually spread fairly slowly through the population compared to random. So the solvers seem to spread this simple information when we see the simple contagion into network faster, and this is probably due to them having more social connections than the non-solvers. But as I've said previously, behavioral spread is not always as simple as just the number of connections you've got, and it might depend on the amount, not just depend on the amount of connections you've got, but the proportion of connections you've got, for instance, to informed individuals. So just going back to that graphic there, which is sped up massively. I don't know why it's sped up so much, but where you've got the same start points of two different contagions. So you've got a simple contagion, which is in blue, and the complex contagion in red, both starting at those two individuals in the middle there, but then spreading in totally different ways throughout that network, based on one of them spreading on the total number of connections to informed individuals, and the other one spread in where individuals adopt a new behavior as it's when they've got a higher proportion of connections to informed individuals. And then what we see when we simulate a complex contagion is we see the actual opposite spread. So in this case, now the solvers actually spread these sorts of solutions slower than the non-solvers. So we've flipped it around, and it's because even though they've got more connections, they've got weaker connections and less clustered connections, which means that as they come up with a new solution, that might be a complex contagion, it spreads slower than what we'd expect at random. On the flip side, the non-solvers would spread these sorts of complex contagions faster than what we'd expect at random. So the pattern's reversed, and the solvers spread the complex information slower than expected at random. So then this is where we can go back to this prediction that more connections will mean that individuals are more likely to acquire new information, and say that that might not actually be the case in that context. And then we also decided to look at this in a more simple context as well, rather than thinking about individual start points in the network in relation to whether they were found to be problem solvers or non-solvers in the lab. We wanted to just assess how individual social network positions shape different types of behavioral contagions. So by measuring all on a local scale. So by measuring all these social networks across these years, and then looking at within each site, what is a particular social network within each site, within each weekend, we can create a lot of replicated social networks, and then we can start simulating different types of social learning strategies on top of these networks to say how might different types of contagions spread. So first of all, we'll simulate a simple contagion, and then we simulate 3 different rules of complex contagion. I won't go into them: threshold, proportion, and conformity, which they're all in this paper if you guys want to look it up in a bit more detail. And then the question that we ask then is simply, what's the correlation between the order of acquisition, so how likely individuals are to acquire this particular contagion, and their network metric. So we calculate everyone's real world network metric, what's their clustering coefficient, what's their degree, what's their betweenness, and saying how's that correlated with how likely they are to get these different types of contagions once we start simulating them through these localized networks. And we repeat this 100 times for each different learning rule at each different site. So what I'm going to plot here is, first of all, just thinking about simple contagions, where we've got the order of acquisition on the x-axis there, and then individuals' weighted degree on the y-axis there. So this is individuals, how quickly they get that particular piece of information, and this is their weighted degree, so how social they are on the y-axis. And we can plot a different line for every different network size that we see. And we see when we look at a simple contagion, what we expect as individual centrality declines. So when individuals have got a lower centrality, they're more likely to acquire this information later on than individuals with higher centralities. So we see this typical pattern as what we'd expect. The more central you are, the more likely you are to acquire the information faster. The less central you are, the more likely you are to acquire this information later down the line. Whilst when we look at a complex contagion, in this case, I've just shown the threshold rule here, we see that this changes actually quite dramatically, especially for larger network sizes. But we see it's no longer the case that individuals who have the highest centrality are the ones who get the information early on. They're not the first in the order of acquisitions. But we actually see it change so that individuals of the highest centrality might end up acquiring these pieces of information later, around number 30 or something like that, compared to those with low centrality. So it really changes the shape so much that actually, if you look at the correlation coefficient between individuals' weighted degree and how quickly they acquire some information of the complex contagion part, there's actually no real correlation anymore, especially for the larger network sizes. So again, this sort of gets rid of the idea that more connections are always going to mean that individuals are more likely to acquire new pieces of information, particularly when you're thinking about these complex contagions. And this is just a little plug for if anyone's got a social network and they want to simulate a different types of contagions on top of that. There's a package online now which you can use on Archive, where you can just simply go on it, plug your network in, or simulate a network if you've not got one, and then tell it what type of contagion you want to simulate on top, and then assess all different things like how individuals start places in that network might shape their contagion, or anything else you might want to look at. So finally, if we've still got enough time, just quickly move on to how we use our system for experiments for testing social contagions as well. So the observational methods are great. You know, we're trying to, regardless of what system we're working on, we're trying to record interactions and then deduce some sort of social network and then compare it to some sort of social process. But even with the most intensive recording you can imagine, it's still very difficult to disentangle correlation from causation. So ideally, what you want to be able to do is manipulate social interactions directly to modify the social network in some way, and then actually directly test what does this mean for these social processes. So this is what we developed, a feeder which looks exactly the same as the other feeders, but it's got a clear flap in front of the feeding hole. And then based on an individual's RFID tag, we can control which individuals can access which feeder type and who else they can access it with. So we can actually shape which individuals can forage in flocks together. So here, this is an example of all these birds are allowed to forage at this particular feeder. So as they land, it reads the tag and it decides whether or not they can feed there. This is an individual who we read his tag, but he's not allowed to feed there for whatever reason. He's fallen in the group of individuals that aren't allowed to feed at this particular feeder. He's got to go elsewhere. And this is what it looks like. They'll have a little peck for a little bit and then fly off. But they soon learn which feeders they can and they can't go to. And in that way, we can shape their foraging flocks. So one question we've asked using this approach is, how does segregation in social networks carry over into the transmission of information? So if we segregate individuals out, how does that carry over into transmission of information? And the way we did this, one of the most simple experiments we've done using these approaches, is we take every individual's pit tag, and they've all got a random number assigned to them, and then we split the population up by odds and evens. So odd birds with odd numbered pit tags can feed at these feeding stations, and birds with even numbered pit tags can feed at these feeding stations. So they're all still spatially associated. This is a very small distance for a bird, but then socially segregated in terms of which flocks they can forage in together. So we segregated the birds using these selective feeders, but then we placed out new open food patches, which any birds could access. And then we monitored the discovery on these and then used network-based diffusion analyses to understand how the information is spreading through the network. And what we find is that individuals learn the location of new open food patches through whichever associates we've imposed upon them. So these manipulated networks carry over into how information spreads. And we also find they value the information of those individuals we've caused them to be with as well. So it might even shape the social learning rules. And then more recently, we've gone a step further to test this idea of how centrality in a social network really does shape social information spread by directly trying to manipulate individuals' own centrality. And then when we do this is we get, so I'll just explain the experimental sites. We'll take a particular site and then we'll assign one site as the high density feeder and one as a low density feeder. So 75% of birds can feed here, only 25% of individuals can feed here. And in that way, you get certain individuals who are highly central in the network, so they've been forced into the central zones, and then other individuals who are now peripheral in the network. So we altered the social density at local sites across the woodland. And then again, we placed out these new open food patches and monitored the discovery of these. And what did we find? Well, first of all, we found that our manipulation were quite successful. So those individuals that we assign as high centrality, it does increase their network density and increases their eigenvector centrality as well. So if we assign an individual as being assigned to the high density treatment or high centrality treatment, it does definitely increase their social centrality in that network. But then when we look at how it alters their chances of information discovery, we see that it's actually very little effects on how it being assigned to being a central bird doesn't necessarily correlate into you being more likely to find new information out about new food patches, and also no changes in the actual order. So it's not, you're not more likely to find new food patches, and you're not likely to be quicker to find new food patches either, which is really interesting. Even though technically that could be viewed as a null result, it does match up with these sort of expectations that we had initially, that it might not just be the case in real world populations that more connections are going to mean you're more likely to acquire a new piece of information or a new behavior, because there's other things at play, such as social learning rules. It's not just how connected you are in that social network. So if we've got a quick chance just to highlight what I think might be some future research questions in this particular area. Something else we're really looking into is, again, using the benefits of the behavioral ecology approach and thinking, how do individuals change across the course of their lifetime as well, in terms of their social centrality, or how likely they are to get a disease or get a piece of information. One thing we've been doing this is in red deer. So this is just a little animation about how much individuals change in their social centrality through their lifetime. This is one deer here, a central deer called Marlin. As she's getting older and older, she's getting less and less connections. And that's what we see with these deers, that they become more socially isolated, more than what you'd expect from just spatial factors and other factors alone. So you can see Marlin getting, it's getting towards the end of her life here, fewer and fewer social connections. I feel like this represents years in my life recently, just losing my friends slowly and getting closer to death. So I can relate to Marlin there. And yeah, so I think some interesting questions coming out of this is, what's the relationship between aging and social behavior? What might this mean for the social structure as a whole? And then how might this carry on into shaping contagions spreading on those social networks? And for anybody interested in those particular questions, there's a free meeting in person, or you can join online, the Royal Society in London, which we're running in February, and everyone is invited. It'd be great to see anybody here there. I'll send an invitation email out, and I'll include all of you guys in the email as well. So it'd be great to see anybody there who can make it. Another factor is, we've considered how simple contagions might shape the spread of diseases and bacteria and things like that. But what about beneficial? What about when the microbe that's being spread is actually beneficial to the host? What might that look like? And a shout out to Aura, who I think has given a fine seminar before, actually, thinking about how social networks predict individuals' gut microbiota. And then more recently, she's been looking at how particular different strands of the microbiota might spread differently in real world populations. And then finally, even though I mentioned this side of behavioral ecology for understanding contagions, it's not really something we've done yet. Thinking about ways of combining lots of different data sets together and then trying to learn from this sort of like a meta-analysis approach about what might shape a contagion, or how might contagions benefit in some settings but be disadvantaged in different social settings. And this shout out to Greg Albury, who started working on these sort of approaches, so trying to amalgamate different social networks, which I imagine some people in here might be involved in this work as well, because it's a fairly large work involving lots of authors. So trying to bring in lots of different social networks, everything we know about density of those populations as well, and then try and draw out some general patterns as well. I won't talk through them, and then finally, one last for further work, and also sort of a limitation on previous work as well, is thinking about how feedback between gaining a piece of information or gaining behavior in particular might shape your actual social position. So currently we've been using this sort of format where information might go into a social network, and we generally assume no feedback in that social network. So individuals pass it on to their associates, and these pass it on to their associates. But what might actually be happening is, as individuals gain a particular behavior, they might become more central. So they might not just have their previous social links, they might actually gain new social links and pass it on to more associates than what we expect. And then these will also become more central and pass it on to more. So thinking about how the contagion itself can shape the society structure, and what that new society structure might mean for the contagion. So yeah, that's why I think behavioural ecology is important for contagions, with an example for the COVID-19 work, and then also particularly using animal social systems for understanding behaviours, and some future research questions there. If anybody's already got the answers to those future research questions, then please do let me know and save us all some time. And thanks as well to everybody, particularly Ben Sheldon, everyone at White and Woods for all the efforts in gathering the data, and all the funding bodies who help that sort of work as well. Yeah, and that's everything from me. So thanks a lot for everybody who made it to the end of the talk and dealt with the Yorkshire accent through its entirety.
Thank you very much, Josh, for such a great talk. It was awesome. And thank you everyone for staying till the very end. I'm going to open the discussion. So I just type in on the chat. If you have a question, just put a question mark. I'll call your name. And then for those of you on YouTube, please type in your question on the YouTube chat, and Lauren will happily read them to us in the Zoom. Remember that there is a delay between the YouTube and the Zoom, so just be patient, but we'll get to you. Okay, so we have our first question comes from Raghavendra, and just as a reminder, we're trying to build a community, so please state your name and where you are from.
Thank you. Can you hear me?
Yep.
I'm Raghavendra Gadakar from the Indian Institute of Science, Bangalore. Thank you. I enjoyed this talk and learned quite a lot, and was very happy to see the connection between behavioral ecology and COVID. I have two questions. The first question, actually, you partly answered in your very last slide. I was wondering about the effect of acquisition itself in changing the network properties of individuals or populations. You touched upon that in your very last slide. But my more specific question is, do you expect differences between this effect of acquiring the contagion? In the first part of your talk, you talked about COVID, where people get disease, they may die or become sick, and they get information. So is it likely that there is a negative effect of acquiring a disease and a positive effect of acquiring information? So would there be differences between different kinds of contagions in their effect in modifying the network properties themselves?
Yeah, it's as you say, it's a really super interesting area with a lot of very interesting questions like the ones you raised there. And I think your question about will there be different effects of that feedback based on the type of contagion is something that we really need to answer, because if we could answer that, it'd really help with simulating these sort of things rather than just having to measure them. So if we did know particular types of diseases always reduced social behavior by X number of connections in an ideal world, for instance, or if we know actually gaining new pieces of information always increase social connections by 2, then you can actually start using those parameters within your simulation models so you can actually predict how diseases might spread or behaviors might spread in a much better way than we currently do, because obviously currently we're stuck at not including that parameter in the simulations. So until we've got that sort of detailed knowledge, we can either guess at it and say, oh, well, maybe there's some sort of feedback between the contagion and them likely to become more socially isolated as they start displaying the disease or something like that. Or you can try and measure it on a small scale and try and plug it in. But if we gain some sort of general understanding of that, which will probably come from thinking about these things across different databases. And I think animal databases is great to do that, because we've got so much variation in what those societies look like. So if we can come up with a general rule that works for across animal societies, I think it'll be easily applied to human societies, because they generally look fairly similar compared to looking across different species, for instance. I think that'd be really useful. Yeah, and I do include the feedback slide as my last slide as a disclaimer, because it does, it does, if you're not considering that feedback, it does affect what sort of weight you can put into your predictions from, even if you've got the real world social network, and even if you know everything you need to know about our particular contagion spreads, if you put those two things together but don't know how they interact, your predictions are going to be wrong. So those sort of things.
Wonderful. In this context, I think it will be especially interesting to think of disease-causing contagions, which actually modify the behavior of the host to benefit the spread of the disease through affecting the network properties of the individual. So disease may not always negatively affect the spreading properties of the individual, but might actually enhance it. And this could be a very interesting aspect to look at, the effect of the contagion, the selfish interest of the contagion. So fascinating. I have a second question, which I actually had it even before your talk when I read the papers you had circulated. So in your 2020 Nature Medicine paper, your last sentence says, our findings suggest that targeted tracing and quarantine strategies would be most efficient when combined with other control measures such as physical distancing. This is a kind of qualitative statement, not very surprising. The real interest would be if you can actually prioritize these or give quantitative numbers on this, which is how much important is which. Because if you combine all of these things, they must work. I mean, that's not very informative. But if you can say this is more important than this, if you can untangle these, disentangle these, and then put priorities or numbers or effectiveness on this, that would then just be absolutely wonderful, especially in the human context.
Yeah, definitely. Yeah, again, agree with that. Yeah, it's quite a qualitative statement. But I suppose one of the messages we were going for with that was, rather than just going to the extreme with one approach, like contact tracing, for instance, going to the extreme and doing things like secondary contact tracing, the amount of effort to do that sort of stuff and the social disruption that it caused, we did show quantitatively that you'd be better off just doing a little bit of social distancing and a little bit of contact tracing. If you did the contact tracing, what we also didn't know at that point is how badly the contact tracing was going to be done in the UK. So we had no idea. So it fell apart in terms of the actual practice of that. But yeah, when we were modeling it with a certain efficiency of being able to do the contact tracing to a particular level. So yeah, and even though we did make that qualitative statement, yeah, we did try and show quantitatively that combining a few interventions on a smaller scale would show a greater impact on controlling this particular type of disease, these particular types of social networks. And then that's sort of, we also put that shiny app together there that I show where you can, test yourself. You can say, oh, what happens if we do maximum amount of contact tracing, but no social distancing, and you get a spread estimate for that compared to what about if we just do 10% social distancing and 10% contact tracing. So you can play around and have a little look. But yeah, I think the gold standard would be trying to really say, even across different systems, what would be the optimal approach given this different disease, and how general is that for if you're looking in different social contexts or different systems. That'd be really what you'd be trying to do, and not something that we've managed to do yet.
Thank you. That was a lot of fun. Thank you.
Yes, thank you.
Thank you. Okay, so next question comes from Eduardo.
Thank you, Adriana. I'm Eduardo Fernandez Duque from Yale University. Thank you, Josh. Very, very interesting talk. I have a very simple question. I'm curious to learn some more about the natural history of the species. So you share with us, it's always fun to learn about the extremes, and you share with us the stories of those animals who, if I remember well, had like 65 chicks or things like that. For animals that, if I remember well, some of them may live 9 years. So actually, it's a couple of questions, very concrete. So for animals like that, they're really producing, say, 7-8 chicks a year. I'm wondering how that happens, if it happens different clutches, different partners. And the second one is, what happens at the other end of the distribution? So what kind of shape does the distribution, what's the extent of skew, irregular skew in the species?
Yeah, so yeah, it's quite a big shift, but actually so even though I talk about the extremes in terms of the oldest birds and the ones who laid the most eggs and things like that, the other extreme of that is obviously the eggs that never come to fruition. They either fail as an egg or die as a hatchling, or never make it out of the nest as a fledgling. And that's the large majority of birds in our population as well. So most eggs don't turn into a bird. And then also most birds that do actually fledge out of the nest, also we don't actually see again. So there's another level of cutoff there as well. So most of the fledglings don't recruit to the population as well. Some years it ends up being Slightly more than 50% will recruit, but in most years, if we take an average, most of the fledglings out of the nest don't recruit. So there's a lot of mortality early on in these birds' life. If they manage to make it to, say, 2 years old, then they do normally make it longer till. So if they make it out to the first year and manage to start breeding, and we call that 1 year old, say, then they will normally manage to make it longer and can live, 3 or 4 years is not surprising. That ends up being sort of a median for those birds that live a bit longer, but most are going to die early on. So they either starve to death in the nest, which happens a lot because the great tits need 2 parents to raise the chicks as well. So if there's a divorce in the middle of the season, or if one of the birds is killed by a sparrowhawk, which happens a lot as well, or if conditions just get too harsh and one of the birds decides to abandon the nest, then all the chicks die. That's definite. And then also once they come out of the nest, there's a lot of sparrowhawks in White Woods now, which kill a lot of our chicks. And then they also, in the cold winters as well, we see a lot of seem like they probably starve to death as well. So the cold and wet winters often result in a lot of birds, a lot of juveniles going missing as well. Seems like they don't make it through. So social information in terms of food, especially in that first year of life, is probably very important to these birds in that context. And then also avoiding predators. Again, it seems like from looking at which sort of birds sparrowhawks are likely to take, they are very likely to take the youngest birds who are less maybe escaping or maybe avoiding predation areas in the first place. So it's the young birds that are likely to get hammered by predators and then also die of starvation, either in the nest or after the nest. So it's a hard world for a young great tit in White Woods. But then you do get those other extremes as well, where we see certain individuals, especially because of the number of individuals we've monitored. We can see the happy endings as well as these, these great grandmothers who lived for 9 years and produced lots and lots of fledglings over their life, and that happens by so they'll breed each year, and especially these ones that we've have recorded, say, more than 50 nestlings from, they would have bred every single year of their life. Sometimes with the same partner, sometimes with a different partner. So the divorce rate is about 50% in the population for those pairs that persist to the next year. So about 50% divorce rate. And we do see differences in individual level quality as well. So those that breed good 1 year are also likely to breed good the next year. Those who fail 1 year are also likely to fail the next year as well. So yeah, it's a really sort of interesting species to work on, and very easy to work on as well. So that's something else I should tout for the great tits. They're very happy with disturbance and all the different experiments we throw at them, and any sort of food we put in the woods, they're always happy to come and have a look, or a little puzzle device, they'll always come and have a little gander. So yeah, great species to work on.
A very quick follow-up. How long does it take you from the office to the field site? So how convenient is the
so now we've got offices in the middle of the field site as well. So you can literally be in the office and in the field site at the same time. So yeah, even if you're coming from Central Oxford, it's 10 minutes from the university to get to White Woods. So by car, or similar by bike as well. So yeah, it's great. And then we've got a field station just outside of the woods, and then also recently done some offices in the middle of the woods as well.
Thank you very much.
Thank you, Lardo. So I'm going to skip myself, and I'm going to call Sari.
Hi, I'm Sari van Belle. I study black howler monkeys, and I'm at the University of Texas in Austin, but I'm actually located in Mexico. I had a question about the models, the network models, or how information moves through the network when one individual is seeded with information, and whether that individual is either a solver or a non-solver. In that those two different types of individuals, like the way information spread through the network, seem to kind of be just the opposite of one another, kind of mirror images. And that, I don't know, seemed to be related to how, because it was measured relative to randomly spreading, or how information would spread if it was random, but there is little information of what you mean by random. Is it that you randomly choose a solver and then randomly a non-solver, or that you choose either a solver or a non-solver randomly. And so these two patterns level them off, and therefore they need to be mirror images, so that if the solver is faster than random, then the non-solver has to be slower because it's relative to that random pattern. And then it was interesting that this became reversed if it was a more complex network. Could you give a little bit more information of how to interpret that data?
Yeah. So yeah, in terms of the actual simulations themselves, that even though that paper's not published yet, the paper in eLife that were referenced there gives some more details in terms of the exact mechanics of the simulations. But yeah, and then in return to the question of which individuals are you seeding, what do you mean when you say seeding a random individual? In this case, it meant seeding either a solver or a non-solver or an unknown individual. So there's a lot of individuals in the population who we didn't have the solving phenotype of as well. So we got, say, 500 of the individuals in those social networks we knew the solving types of, but there were probably over 1,000 individuals in the network that we didn't manage to capture and assay for problem solving ability for whatever reason. So yeah, random refers to seeding a random individual in the network, and then obviously you want to figure out, okay, if we just take any individual, what's the average rate of spread? So we do that loads and loads of times, and obviously because of the stochastic nature of these simulations as well, just where you start in the network doesn't determine how that's going to spread, because we've got some probability elements in that spread as well, so it can differ a lot even with the same start point. So you want to do that thousands of times to get sort of an average rate of random spread. And then so the lines I plotted there were the average spread rates for seeding solvers. For instance, if we just imagine one of those red lines, that's the average for that particular year. When you choose a solver, that's the average spread rate for that particular year. And then, yeah, as I said, then it's like almost by necessity that the non-solver, it doesn't have to be, because there is a lot of individuals who aren't sampled for solving ability, but it's very likely then that non-solvers are going to differ from that red curve, because they're more than what you'd expect at random. So yeah, the idea was to try and grasp, not really to try and get an idea of the actual spread rate per se, and that's why we do it spread rate compared to random, but how the type of contagion might shift which individuals are important to that particular spread rate, to try and demonstrate or test that prediction that those problem solvers, even though they might be more likely to have more social connections for whatever reason, we don't know yet as well. That's another big unanswered question, but that'd be really great to go into in more detail, especially if anyone's interested in sort of cognition processes in general. It'd be good to know what people think about that. But yeah, to think about if that phenotype of being a problem solver means you're more likely to be socially connected, does it also mean if you come up with a solution, it spreads faster? And the sort of answer to that is, well, actually, if it's a complex contagion, not necessarily, because it doesn't just depend on the number of connections. But yeah, it'd be great to explore that sort of question with different social networks going forward as well, rather than just the great tip one.
Thank you.
Cheers.
Thanks, Barry. Our next question comes from David.
Hi, folks. Thanks, Josh. That was really interesting. Yeah, I'm David Fisher. I'm at the University of Aberdeen. So there was a really cool preprint out recently which looked at did a meta-analysis kind of approach across lots of different animal networks. And along with answering their own research questions, they mentioned that between 2/3 and 3/4 of all the variation in network structure was among studies, which kind of implies that there's not much left to actually explore any interesting biology, because its choices of study methodology are actually accounting for the vast majority of variation in networks. So we often like to say that everyone using social networks allows us to kind of better compare what we're all doing and so on. But like, given that variation, does it? And do you have a cunning plan for how you're going to compare across networks?
Yeah, well, I don't have all the answers if that's what you mean, but I've not actually read that paper yet, but I do need it's on the top of my reading list. It looks really interesting, and yeah, that's the first I've heard of that particular finding. That's a really interesting finding as well. But when we're comparing networks, I suppose it's not surprising, and epidemiologists would agree, that networks in different sort of social contexts, especially if it's between different species, are going to be very different, and then account for a lot of the variation, just which sort of system you're looking at. it's going to be hard to draw similarities there. And that's why I think these permuted population approaches can be particularly good in the comparative context. So what you ideally want to do in each sense is say, okay, so rather than saying, let's take a really simple metric like what's the variation in individuals' network positions in this particular network, and then comparing that measure, call that measure Y, to the same measure, what's the variation in individuals' network positions in network B, for instance, to network A. Those two metrics are likely to be very different simply because they're from different systems, they've been measured in different ways, blah, 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 blah. What I think you're better off doing is for each instance, take your first network, you take network A, and you say, what's the variation of individuals' network positions, if that's the measure you're going for, measure Y, and then you say, rather than just using that as a raw measure, let's see how that differs from what we'd expect at random, given the number of individuals in that network, how that network was monitored, everything else we know about that network. So generate a load of permutations of that network and then say, okay, what's our measure Y now? And not in terms of the actual raw measure Y, but how different it is from itself, from its expected, given its structure. And then compare that to when you go to network B, do the same with network B. Instead of just using the raw metric Y, you want to see how different it is from what you'd expect under these different circumstances, and then compare them two things together. And I think that's where we'll get somewhere with the sort of meta-analysis of networks, by rather than just using the raw metrics or trying to calculate some metric over and above all of them that sort of fits, is to say how different are they expected from themselves, because then we capture lots of things like number of individuals, number of associations monitored, all that sort of stuff anyway. Given that, what does it look like at random? How different is this over and above? And then hopefully we're getting some aspect of the actual meaningful behavior that we can compare across. Again, that's kind of that's some thoughts off the top of my head, but yeah, I think that's the way to go.
Yeah, basically generating effect sizes. I've thought of some, not with randomizations, but I've wondered about something similar with some modeling approaches. So
yeah,
that could be cool.
Good area for going forward though, and yeah, interesting to see what people will do with that. And I think again, animal social networks is a perfect place to do it.
Great, thank you, David. And our next question comes from Clara.
I guess you mean Clara?
Yes.
Okay. I'm Clara B. Jones, retired social evolution. I have one brief comment and then two questions. Don't forget that you can conduct experiments with simulations, particularly agent-based modeling. And so you can do experiments with human data, data on endangered species, et cetera, et cetera, et cetera. My first question is, let's see. I'm wondering if this is not just an intellectual exercise, how would you relate your findings to, say, the evolution of population structure or the evolution of groups or something related to social evolution?
Yeah. Yeah, that's a really good question. I suppose the context I've thought most about that in so far is just within our particular great tip population, because there you've got some sort of ability to look at changes across generation in terms of individuals' social phenotypes and which individuals' social phenotypes might relate to positive fitness outputs or detrimental fitness outputs. So in that context, in thinking about a single system like our great tip system, where we've got individuals of known social network positions, and we can calculate a social phenotype on that, those individuals, you know, ideally what you want to do is, first of all, is think, well, is these social phenotypes that we're estimating in the social network heritable to any extent? You know, so can we estimate some heritability to that social phenotype? And then also, is there some sort of fitness advantage of being socially central or being social peripheral, whatever it ends up being in terms of likelihood of survival, which we've got for these birds as well, and number of offspring that we've got from these birds. And then, you know, what we've not done yet properly is try to think, okay, what's the relationship between individuals' genetics and their social phenotypes? And to do, I know he's interested in that sort of stuff. Get someone like that involved. We've played around with it a little bit, and it looks like they definitely are heritable, and there's definitely some indirect genetic effects of these social phenotypes as well. And we also know that there's some fitness consequences. So, like our preliminary analysis show that in general, social centrality, no matter how you measure it, is related generally to higher fitness outputs. But the way that we've fallen down so far is there's so many other covariates of fitness and social centrality that go together hand in hand. For instance, in certain parts of our woodland, it's very productive parts of woodland, which means a lot of birds go there because it's very productive in the winter, but it's also very productive in the spring, so that once they go there, they're more likely to breed there, and they're more likely to have better outputs because it's better to be a breeding bird there as well as a breeding bird through winter. So automatically you get this sort of spatial covariation between where individuals go in the winter, how many individuals are there, where individuals are in the spring, which other individuals are there, and their breeding output as well. So what we've never managed to really nail, because we're using observational data probably, is to get those relationships over and above what you'd expect from our spatial structure. And that's where we tried to get a grant for this, but we didn't get it funded. The idea was to actually manipulate individual social network positions over quite a few years. So we force them into peripheral positions or force them into central positions, and then test the impact of that on their fitness, and then try and relate that to the observational data. But yeah, we didn't get that funded, so we never got to that. We've played around with the observational data, but never really nailed it in terms of an experiment.
Yeah, the fact that you have individual data on individuals is really interesting. And then a quick question, and my memory could fail me here, but haven't the epidemiologists found that there's kind of a general pattern of diffusion, first exponential and then asymptote? Are you finding that? Do you expect your data to be different?
Yeah. So in all the human populations that we work on, we find that same sort of pattern, and that's the sort of pattern and the parameters that we work around as well when we're trying to say different from expected or higher than expected. And then also when we're thinking about mitigation methods, you want to mitigate it before you see that sort of explosion and things like that. So with the human data, but I think, yeah, interestingly, the animal social network data could end up being very different to that, especially when you think about social systems like lion prides, for instance, or you know, these sort of like really strong family group systems, primate systems, for instance. They might be very different to the human communities that people have worked so much on to think about the shape of these slopes. It might start changing things a lot, or even animals that are very asocial in general and just have a few infrequent social contacts, or might be their social lives might mainly be shaped through who to avoid rather than who to interact with. Those sort of systems might give a total different shape to that, which might, when we're thinking about contagions, might mean that we've got to change some of the ways we initially parameterize those models, definitely.
Thanks, great answers.
Thanks.
Thanks, Clara. Our next question comes from Christina.
Hi, good morning. Thanks for the talk. I'm Christina Nord. I'm a postdoc at the California National Primate Research Center, and I'm super interested in social learning and cultural transmission and on those kinds of things. And I'm wondering if you thought about, or what your work can speak to the proximate mechanisms involved in how behavior spreads. I think one of the most, the critical decision in something like network-based diffusion analysis is which network to use to look at for spread, because it originally was used for things like grooming networks or proximate networks and looking at how behavior spreads through those networks, but that doesn't really tell you exactly how it's spreading. And I think that's really crucial in things like knowing what kind of mitigation factors we need for things like viral contagions. You know, should we mask, or should we socially distance, or should we hang out outside? Or even for behaviors, how is it best to teach, children exactly what is the proximate mechanism going on there. So can you speak to kind of how these, it seems to me that a lot of the social learning literature, social contagion literature is more of a proxy of kind of how things are going on. It isn't actually getting at to like what's going on the ground.
Yeah, that's a really interesting question, and some that well, the general sort of topic of that caught my interest a few years back when I first started using network-based diffusion analyses. That sort of concern first came to mind as well, that, oh, wait a minute, these models are all based on the idea of the raw amount of connection you've got to other individuals influences your chance of gaining that piece of information. Because often they're done with information, these network diffusion models. And then whenever they're calculating that S parameter, that vital S parameter to say, okay, how much social learning is going on, is all based on the assumption that it's the raw amount of social connections between individuals. So however much time you spend with informed individuals directly relates to your likelihood of gaining that piece of information. And through that assumption is where you gain the S value by saying, okay, well, so what's the actual shape of that? And that's your S parameter. So yeah, and then reading that, and then at the same time reading the sort of sociology literature, that information doesn't necessarily spread based on how much time you spend with informed individuals. You know, even if you spend 10 hours with an informed individual, but you've spent 100 hours with individuals not doing that behavior, you might be less likely to take on that behavior compared to an individual who's just spent 1 hour with an informed individual, but that's the only person they've hung around with. So then, like you said, there's no sort of like actual idea about how these things are spreading. It's all just based on total amount of connectivity to informed individuals should relate to increased chance of spread. But that's not always the case. And that's why then we will hop it. I put together this complex MBDA analysis package where you can actually say, either in the terms of, if you've got a real world social network and you've got a real diffusion on top of that social network, you can take that package and you can say, okay, what's the general social spread? If we use a normal MBDA model, how much do we think of this spread is due to social learning? And then you get your S parameter. But you can also say, wait a minute, what about if it's not just that really simple rule, but there's some other rules going on? So then you can choose a rule. You can think, oh, actually in my parameter system, for instance, I think there's a threshold rule going on or a conformity rule going on. If we put that into the model as well to say, now our expectation isn't that it's the total amount of connectivity you've got to informed individuals, it's your proportional connectivity to informed individuals compared to you've got to uninformed individuals. How does that relate to, first of all, the S parameter? So does that show increased or less social learning? And also, is that model a better fit than a very simple model of just total amount of connectivity? So I think from those sort of modeling approaches, you can kind of get at, well, let's plug in a different social learning rule and see if that explains the spread better or worse than if we don't have a particular social rule in play. And yeah, I think it's, as you mentioned there as well, I agree that the social learning sort of literature and the social contagion literature have been fairly separate. And it'd be really nice to see people think, oh, we've found evidence of all these different social learning strategies in a population. So rather than just treating like social spread as epidemiological models, just taking them out of whatever, some human disease spread modeling situation and applying it to our network, let's think about the social learning rules we've seen in animal populations and apply them when we're thinking about how these things might spread. And that's why I think it's really important for people to be on the ground studying, you know, if you're working on a particular animal system, not just looking at the connections between those individuals, but how do they actually interact? How do they learn off each other? You know, the only way you're going to do that is through looking at specific assays and thinking, oh yeah, there does actually seem like there's a social learning rule at play here, or there's a particular sort of social preferences going on here, and then shaping that into your models. Otherwise, you'll miss a whole lot of interesting social stuff, and you'll also predict, if you try and predict from those models, you'll get it wrong as well if you don't include those things. So I think, yeah, the proximate side of stuff is really interesting. Probably more to do, also interesting stuff to do with the start points in social networks as well. Like there, I was just really simply saying, oh, we might have some solvers and non-solvers in our population. How might that relate to spread? Without actually ever saying what makes an individual a solver or a non-solver in the first place. You know, understanding those sort of things, there's a lot of work to be done there.
Thanks very much.
No, thanks. That's great.
Thank you, Christina. And I don't see any other question. here. So I'll take my question, and I think there's no question in the YouTube, right, Lauren? Okay. So I have two questions. One is basically a follow-up on Christina's, and I was thinking about, you know, all the kind of like history of all these birds, the gratids. So how is the fitness consequence of the solvers versus the non-solvers in your population? Do they actually do better?
Yeah, so the solvers do in general have higher breeding success than the non-solvers, which as a phenotype is quite interesting. But then the interesting part of the solving behavior is it looks like it's spatially segregated as well. So you get certain area of the woodlands where a lot of the solvers go, and those also seem to be the fairly high productive areas as well of the woodland. So it's hard to really test the causal link there. But you know, whether it's the solvers are better at identifying high quality areas as well, you know, so maybe they end up in the high quality areas by means of being a solver, problem solver, finding the best places and then having higher breeding success. Or it might just be a correlation that for whatever reason solvers end up in these high productivity areas, and then we see this covariation between them. So yeah, it's really hard to disentangle it. What we really want to do is we want to make different groups of either solvers or non-solvers, so actually change the network in relation to, well, both the spatial structure and also the social structure in relation to those phenotypes. So in a perfect world, we'd be able to carry out some more experiments where we assay birds' problem-solving ability and then choose where they go, so where they can feed, which parts of the woodland they're going to be hanging around with, and which other birds they're going to hang around with while they're there, based on that phenotype. And then we can really look at what the fitness consequences directly, but also directly go back to, okay, well, does it actually causally change the spread of any new innovations in that particular society as well? Or how does it change the network structure? Do we see when we force a load of solvers together, because we know individually in a network they've got more social connections, do we see a really highly densely connected network? If we take the non-solvers, do we see just a network made up of a few strong bonds instead? So to try and get at that sort of thing, it'd be brilliant to do some more experiments where we can start separating individuals up by that problem solving status, both for the fitness consequences like you mentioned, but then also for thinking about how things spread in these networks and the social structure as a whole.
Can you, how do you say that, move the solvers to a non-productive area, like translocate them totally, and then just like see how they do?
Yeah, the translocations don't work very well because they're quite good at going where they want to, like quickly, if you put them in a place where they don't want to be. But what does work is sort of like teasing them into a particular area. So if you put a feeder sort of in a certain position where only the solvers can access it every weekend, or if you have it a few days a week, or have it for a little amount of time, and then base them there for like a short period, you can kind of get them going to that particular place based on which feeder they can access, and then leave them there afterwards. So you can tease them into a particular area. It's hard to force them into it in the wild, but you can sort of lure them in and then hope that they stick around. And you could definitely, because there's so much variation even at a small spatial scale, you can easily force individuals to areas which are generally more highly productive or lower productivity, or even social settings like more central or more peripheral, is what you can imagine doing fairly easily, and then seeing how that carries over into other parts of their life as well.
Okay, I see. And my previous question was about the size of the networks. So, I mean, pretty much all the examples you show, they are large networks, right?
Yep.
Then at the very end, you were mentioning like the size of the network, and you started like at 20, say 20, it's a small network, but we see some animal groups that are in even smaller groups. So is it like we should not, or would the predictions in small networks be that everyone would get sick, and we shouldn't care about the social structure in the small networks, or how does it relate or interplay between the network size and the actual contagion process?
Yeah, that's definitely a really like complex question, because trying to understand how the network size might change those contagion process is a big area, but an important area. And something, so yeah, as you mentioned there, we went to like sizes of from say 20 to 60, and that's because with that particular study, we were interested in that question as well. We thought, how might the local social environment in that simple measure of number of individuals around shape how these contagions might spread over and above individuals' network positions, with a sort of general idea of, if you're really socially central as an individual, but only in a group of 10 individuals, you're not going to be that much different from an individual who's quite peripheral, because you've only got a maximum connection of 9 anyway. They've got probably 3 connections. So there's not much difference compared to if you're in a big network, you're very socially central, you're in 60, there's 60 other birds around in that particular area. So we split all the woodland up into different areas and made social networks based on each site for that study to do this. And then you can see this massive variation between how socially connected individuals are. And yeah, and the cool thing is you do see a lot of the variation in how contagions spread, both in terms of the relationship between how central an individual they are and their likelihood of acquiring a piece of information, but also the difference between, say, a simple and a complex contagion, how those spread does relate to the network size, as you guessed there. So you see, as you go to these bigger network sizes, there's more of a difference in, well, within our species at least, there's more of a difference in the different types of contagion. So it's as you go to the sort of more dense sites, more individuals around, where a complex contagion is going to spread more differently from what you'd expect through a simple contagion. How that might differ in those smaller networks, for instance, like there's been some work done on fairly small human groups as well to show that clustering coefficient in small networks becomes really important. You know, so if your best friend, if you've got 2 best friends and they're also best friends with each other, then that can make a big difference to how ideas flow, compared to if your 2 best friends don't know each other. So rather than just going for total number of connections, you can start looking at those more sort of clique measures to think about how it might shape contagions. And yeah, Damon Santola's work has done some fairly small groups as well, probably similar to a lot of like the primate studies, between 20 and 30 individuals, and showing that even in those sort of small networks, you can see fairly big impacts on on spread based on what contagions at play or what sort of social rules at play. Oh, and also I suppose that figure, that theoretical figure I show in the thing of the blue contagion and the red contagion spreading differently, that's only 24 individuals. So the network's not massive, but you still see, and even though that's a toy example, it is based on the rule of only spread to the either the most connected individual or to the highest proportion connected individual. That's the difference there. So you can, you know, even with small networks, you can predict a fairly big difference based on the structure and what type of contagions at play. But yeah, overall, our network stuff in our great tits has been based on probably bigger networks than what most people would use, and it'd be good to see some similar stuff in smaller networks to see how well that translates to those areas as well.
That'll be great. And also in like third networks, like the sparse networks, where you don't have like everyone each other and everything, because that's also changing the actual way of transmission. But okay, I'm going to stop there, and I'm going to give the word to Lauren, and then I'll go with you, Eduardo. Is that okay? Good.
Hi, Josh. Thank you for a really fascinating talk. So for everyone, I'm Lauren Hayes. I'm University of Tennessee at Chattanooga. I study social mammals, focus on degus in Chile. And so, Josh, I'm really interesting, and it just got my mind going. I'm not sure I can really formulate a great question, but I had these thoughts in my head, and I wanted to see what you think. So first out, are these in the great tits, are solvers more likely to hang out with solvers than they are non-solvers? And would that affect, so I'm getting at the idea of homophily, would that affect sort of information transfer, disease transfer?
Yeah, you're right. They are more likely to hang around with, so the solving phenotype does determine assortment in the network. Well, it does relate to assortment in the network. So the solvers are disproportionately connected to other solvers, and the non-solvers are disproportionately connected to other non-solvers as well. So there is assortment by that trait. But what there's not, or what there's not significant evidence for, is that is over and above what you'd expect from differences in social phenotypes. So there's this phenomenon in social networks where most social networks end up as assorted by degree. So the more connections you've got, the more likely you are to be connected to other highly connected individuals. And that kind of rise just by how we monitor these networks and how these networks form. So that's a common sort of phenomenon across these networks, assortment by degree. So assortment by social popularity always ends up happening. And then because the solvers are more popular or got more connections than the non-solvers, I suppose it's not surprising that you see assortment by this phenotype as well, because it's correlated with it. And what we can't actually show is that there's assortment over and above what you'd expect from them just being more social. So we don't know what we can't at least show evidence for is that the birds are purposely hanging around with other solvers because they're a solver, or hanging around with other non-solvers because they're a non-solver. But we can show that there's definitely some assortment by social centrality, which then translates to assortment by problem-solving ability as well.
Interesting.
And I suppose then the consequences of that, still important. So if you're looking at consequences for something like spread, then it actually really don't matter whether they're assorting by problem-solving phenotype or social centrality phenotype, because it's the structure of the network that matters. But if you're interested in proximate mechanisms or thinking about what makes these assortment decisions arise and things like that, we're not going to be any help there yet. We need to do some more work. Yeah.
Thanks. And so I guess my next question is really different. And again, I apologize if it's naive. To what extent does stability in the network, and now I'm thinking social stability, like maybe too bad Filippo's gone, because the extent to which fission fusion occurs within a society, how would that influence sort of information transfer in these networks, or disease, I imagine?
Yeah, no, that's another really good question. So you can think about that sort of, it's a nice sort of simple and commonly used demographic measure of turnover in a population. And we can either think about that in our population as a population as a whole, or at each individual site within that population. And each individual site within that population, there's a lot of variation. So some sites experiencing lots of turnover and other sites experiencing not so much turnover. And we see that turnover both on the weekly basis as well, because we monitor every week through the winter, but then also on the yearly basis. So there's definitely a lot of variation in that turnover variable. And what we do know is it relates to variation in social structure as well. So in the ones where you've got lots of turnover. So this paper's not published yet, actually, but it's just been accepted in Animal Ecology. So it should be out fairly soonish. And what we can see in that is that both the number of individuals and the turnover of those individuals relates to network properties. So it shapes the social network as a whole quite a lot. But then what we need to do more of is probably thinking about what you're mentioning there. If we've got a disease or a solution in that population spreading through it, how might that turn over? I suppose some of Lucy Aplin's work has touched on this, where she showed that if you seed a particular culture into different parts of the woodland, and then come back the following year, so seed a particular solution, so you've got to push the red side in the north of the woodland, and the birds have got to push the blue side in the south of the woodland to get to the food. If you do that, and then you come back a year later, even after, say, 50% turnover of that population, you still see that culture's maintained, which is fairly interesting, and suggests you only need a small number of individuals each time to remember and keep that going. So, yeah, I think the turnover will be probably important, but not shape everything. So, again, the nature of diffusions is you probably only need a few individuals each time to get it going. And then you can also think of turnover not just in the context of birds each time, but also across generations, and how much mixing you see across generations as well, if that makes sense. So do you hang around with your parents' kids, parents' friends' kids, or so is there stability across generations where it's the same type of genotypes interacting, even if you've got different individuals, it's similar genotypes interacting, because everyone always hangs around with whoever their parents were friends with, their parents have had kids, and they're your friends now as well. So, those sort of things. What should have done work in hyenas to look at what the class of social inheritance. So is social inheritance at play? Do you generally gain your social connections from who your parents hang around with? And then what does that mean for these sort of effects? Like if you think about disease, then you're going to get the same immunity as your parents. And then if they're buddies have the same sort of strands of disease as what their kids have, then you're going to be getting exposed to that again. Or if you think about something like healthy things like healthy microbiome, you might get your microbiome from your parents, and then their friends will pass it down to their kids as well, and you hang around with same individuals with similar microbiomes. Or behaviors like cultures, it might restrict, if you've got social inheritance, it might restrict the rate at which those new behaviors or ideas can spread, because individuals gain their new behavior from their parents, but they're only hanging around with the friends, the kids of their parents' friends anyway. So it's like you get these local sort of clusterings due to social inheritance. And I think, those sort of things will be really good to explore across different animal societies. For instance, like the hyenas have done it quite well. In our system, we see social inheritance, despite the fact that there's no kin structure. So just by birds choosing to nest in particular places to be around their winter friends means that when they breed, their children are then hanging around with their friends, kids, even though they don't hang around with their parents. So you see this sort of transgenerational effects of social structure, consistent social structure over generations due to social inheritance, regardless of having kin structure. So yeah, really sort of interesting area to dive into, something that we started doing a while ago, but never really had the time or the expertise to do properly. So it would be good to talk to people more about that sort of thing. Definitely. That's great. Thanks.
Well, thank you. Very interesting stuff. Appreciate it.
Cheers.
Eduardo, it's your turn.
Yeah, very quick one. I missed. So you were announcing a meeting? Is that what you're holding in February 24? Can you show us that again? I mean, maybe one could try to get away from the awful weather here to go to the awful weather in the UK.
Yeah, it's going to be great. So this were a meeting that was supposed to take place last year, but then had to be postponed due to UK had large scale travel strikes and also university strikes at the same time. So we postponed the meeting for a year, and it's run through the Royal Society. But my idea was that we're a meeting all about how age might affect individuals' social behaviour and what that might mean for outcomes like contagions or other things as well. So it's going to be a Phil trans issue.
Do you have a link? Can you share? So is there anything on the web that one can browse? Can you post a link on the chatting?
Yeah, let me get that now, actually. And then I'm going to send around an email as well to as many people as I can remember who came to this meeting, inviting everybody directly as well, because it'd be great to see as many people as possible.
Yeah. Usually in February, I tend to look towards the southern hemisphere for traveling, not so much across the ocean.
Yeah, London's not going to be, not as nice as the southern hemisphere, but this meeting will be worth it, despite the rain that you'll definitely get.
That's all I had really. So, but I don't see any other questions.
There are no more questions, and I think there's no questions on the YouTube. So what I'm going to do is to stop the live streaming, and then we can just stay and chat a little more. Thank you, Josh, for your great talk and all this time for the discussion. And we invite you for the next seminar next week with David Kohler. Bye, everyone.